morning. I want to thank ODC for this wonderful celebration of the arts and sciences. I also wanted to tell the earlier speakers how much I enjoyed their presentations. I write extensively about the microbiome, yet I learned a lot of new things. For instance, I did not know that that organoids were being used in microbiome research. So this is the topic of my presentation, leveraging the power of the gut microbes to boost mental health. And I want to start by throwing some questions out at you. Would you go bungee jumping? I guess. Oh. Yeah, I don't either. Would you eat an entire pie in one sitting? No one's messing up about that. Do you avoid social gatherings like parties? Strange as it sounds, our gut microbes influence all of these choices. These everyday residents of the body, as we've learned, are concentrated in the gut. They include bacteria, protozoa, fungi, and other unicellular creatures. Collectively, they're known as our microbiota or microbiome. As we've heard from previous speakers, they aid in digestion, synthesize vitamins we can't make, and carry out many other life-sustaining functions. Most amazing, our microbes talk to our brains. They influence mood, energy level, hunger, memory, perhaps even your personality. If we did a total body census, our microbes actually slightly outnumber our own cells. They are the majority, we are the minority. So viewed from that perspective, it's not so surprising that they would want to say in how we behave. I mean, after all, our own fate is intimately tied to our own. One of the best demonstrations of gut microbes' influence on our behavior comes from studies of these guys. They're called bubble mice, and they are raised in sterile facilities, a protective bubble, if you will, so have no microbiome, no microbiota. If you contrast their behavior to that of a normal mouse, you will notice some striking differences. A normal mouse is a quick and eager learner. If you show it a napkin ring, for example, it's, it will circle and sniff it with great interest. You place it in a maze and it's keen to explore new passageways and remembers where it's been. A bubble mouse could not be more different. It is just as likely to favor the familiar over what's new, exciting, or different. It's slow to learn, quick to forget, and astonishingly, they're just strikingly devoid of fear. For example, if separated from their mothers at a young age, they don't even protest. Even though in a normal mouse, this would be a very traumatic event and lead to lifelong skittishness. Now, if you colonize a bubble mouse with the normal bacteria for that strain, guess what happens? Their behavior normalizes. We're not mice, so does any of this apply to us? Actually, I'd argue a mountain of evidence suggests that it does. So consider this. If you take gut bacteria from an overweight human twin and transfer it to bubble mice, the animals fatten up. Take gut bacteria from a thin twin, transfer it to bubble mice, feed them the same rat chow, and the mice stay thin. Similarly, if you transfer gut bacteria from a depressed person into a bubble mouse, the animal will display depressive-like symptoms. For example, it will give up swimming in a water tank sooner than a bubble mouse that receives gut bacteria from a non-depressed person. That animal, when placed in a water tank, will keep swimming. It will swim longer. It's not so quick to give into despair. So how does this work? How in the world can bacteria alter how we feel and act? They have many techniques, actually, but here's one of their most amazing, one of their coolest. They synthesize hordes of neurotransmitters, hormones, and other psychoactive compounds. Put simply, our gut bacteria can talk to our brain because we, meaning our microbes and human cells, all speak the same language. In fact, some scientists believe this language was invented by bacteria, so they call it microbe speak, and they believe it holds the secret to understanding why bacteria can have such a big impact on our behavior. You may think of bacteria as loners, but in fact, they live in these vast metropolises. As a result, there's a lot of competition. So to defeat their enemies and vie for resources, they need to join forces and strategize. They need to communicate, in other words. And they do this by exchanging chemical messages. Think of it as their form of texting, if you will. And this enables them to keep a running tally of their own numbers relative to that of competing gangs that, may be growing in size or retreating. And when they conclude that, they're larger in number, they will do something pretty amazing. They will all switch on the same gene and something wondrous happens. For example, they all begin marching en masse. Pretty cool, eh? They do this by all producing a slippery soap-like substance that allows them to glide across a surface. For example, in this case, a Petri dish. Or a group of bacteria that is normally well-behaved may suddenly start churning out toxins that they hurl at their adversaries. Or they may all at once begin producing a slimy dome called a biofilm to shield themselves from would-be attackers. Bacteria may even exchange messages to help the host that feeds them. For example, they may act in unison to illuminate their container, turning sea creatures into a torch at night. Incidentally, they do this by turning on the synchronized activation of a gene called luciferase, often to stunning effect. When bacteria come together and act as one, I would argue they demonstrate a collective consciousness, a mind, if you will. And that mind is made up of a collection of signaling molecules that evolved over 3.8 billion years. When animals evolved 600 million years ago, yesterday by evolutionary standards, bacteria likely transferred to them the genes that underlie their language skills. Meet the humble hydra. It's a freshwater inhabitant and one of the first animals to evolve that is still in existence today. As you can see, it's basically a tube packed full of microbes. Note, it's not a very bright fellow. It has no brain. But it does have a diffuse network of nerves over its entire body, what you see there in purple. And that network is sensitive to bacteria and their signaling molecules. Via that network, its resident bacteria are telling the hydra how to behave. In fact, they're already displaying bossy tendencies. The hydra's bacteria regulate the contraction of its body and also the opening of its mouth to feed. After all, they want to get fed too, right? So to pick up where I left off, as animals became more complex, so did their nervous systems. And in mammals, it's split in two, with one branch focusing on internal stimuli and the other on external stimuli. The one that focuses on internal stimuli is called the enteric nervous system and is primarily concerned with digestion. For example, the contraction of the intestine and food cravings. The one that focuses on external stimuli is, sights, sounds, that kind of thing, is better known to us as the central nervous system, and of course includes the brain and spinal cord. Interestingly, the enteric nervous system contains more nerves than found in the spinal cord, and so it's been dubbed the second brain. But if you recall the hydra story, clearly the second brain came first, and despite its focus on digestion, it refuses to cede all control of the animal's behavior to the brain upstairs. Indeed, the enteric system has this high-speed cable that connects it directly to the brain that's called the vagus nerve. And surprising scientists, about 80% of the traffic on this cable is going from the gut to the brain rather than the other way around, as it had long been assumed. Of course, bacteria can also rely on the circulatory system to transfer their psychoactive compounds upstairs to the brain. There's one other very important way in which gut bacteria regulate our behavior, communicate with the brain, and that's by misbehaving. When aggressive bacteria invade the intestinal lining, immune cells rush to the scene. And those immune cells and related compounds, we've recently discovered, do not always stay localized in the gut, but can seep into the bloodstream and be carried to the brain, where they trigger inflammation and depression. Inflammation and depression often go hand in hand. They are a destructive duo. For example, people contemplating suicide often have highly inflamed brains. When you think about it, it's a bit puzzling why the immune system might contribute to depression, but there could be an evolutionary logic to this. When bad bacteria gain the upper hand in the gut, it places an extra burden on the immune system, which has to consume extra calories day after day to contain the threat. And depression, it's theorized, may help us in this effort by allowing us to conserve more energy. So seized by apathy and mental exhaustion, we cut back on everyday activities and often sleep more. So it's theorized that depression allows the body to shunt more resources toward the war effort. Indeed, you can briefly induce a type of malaise that mirrors depression simply by injecting perfectly healthy mice with immune compounds. Despite the fact that they have not been exposed to a single germ, they immediately lose interest in their running wheels, put their heads down, and drift off to sleep. So this suggests that by chronically engaging our immune systems, bad bacteria truly contribute to dark moods and even suicidal impulses. Intriguingly, some microbes may also harm us more directly by eavesdropping on chemical messages our brain is sending downstream to other organs. These bacteria have receptors for adrenaline-like compounds that allow them to sense when we're under acute stress. For example, you just had a fight with your spouse, or you have a really pressing deadline. And when they conclude we're worn down and in a vulnerable state, they may cease cooperating with us and collude to bring us down, for example, by attacking the intestinal lining. Okay. Talk about fair weather friends, eh? Hosts, in turn, quietly listen in on conversations between the gut bacteria living under their roof. Believe it or not, our taste receptors serve as our ear to the wall. You may assume that taste receptors are confined to the tongue, and are solely involved in detecting different flavors. But that long accepted dogma has recently been overturned. Scientists are finding taste receptors scattered throughout the GI tract, and also other tissues, and where their function has nothing to do with taste. Among other things, these receptors tune into the chemical signals that bacteria use to organize themselves for battle. In this way, thus alerted, our bodies can rally the immune system to get a head start in squelching the rebellion. All of this is fascinating, I think you'll agree, but what can we do with this knowledge? How can we use it to live healthier and happier lives? For starters, these insights are dramatically recasting our understanding of treatments for neuropsychiatric conditions and how we might improve them. For example, it's come to light that many common antidepressants actually change the composition of the gut microbiome, encouraging the proliferation of healthy species that help to calm the immune system and dampen inflammation in both the gut and the brain. And based on this finding, scientists are now hoping to create a new generation of more powerful antidepressants that, based on their impact on the gut microbiome. The ketogenic diet's well-established power at inhibiting seizures may also owe its efficacy to the transformation of the gut microbiome. This high-fat, low-carb diet, animal research shows, causes two types of bacteria to grow in numbers in the gut, and neither of these species on their own, but the two together, produce metabolites that raise the level of a neurotransmitter in the brain, and that in turn plays a role in inhibiting overactive neural circuits. So armed with this new understanding of how the ketogenic diet probably works, scientists are hoping to develop better treatments for epilepsy as well. And that could be especially good news for the 20% of patients who do not respond to anti-seizure medicine and really struggle to adhere to this very restrictive diet. As you'll see along the bottom row of that slide, there's a lot of foods that are prohibited on this diet. A little known but very promising treatment for a variety of neuropsychiatric conditions is called vagus nerve stimulation, or VNS, and it too may have a microbiome-based rationale. As its name suggests, VNS entails electrically stimulating the vagus nerve via an electrode that's implanted in the chest. Although this therapy evolved independently of microbiome science, some scientists think that the current may actually mimic the effects of gut bacteria on the nerve. Now that's speculative, but what we do know for sure is that VNS strengthens the gut barrier, preventing pathogenic bacteria from breaching it, and then triggering an inflammatory response that spreads to the brain. Encouragingly, the FDA has already approved VNS for the treatment of epilepsy and depression that does not respond to standard therapy. Even more exciting, clinical trials suggest that VNS could also benefit people who suffer from PTSD, ADHD, and OCD. The use of powerful new weight loss drugs like Wegovy and Saxenda has skyrocketed around the world. I wouldn't be surprised if a doctor next door in the Gangnam district is prescribing them. And exactly how these drugs help to regulate our food intake is not fully understood, but it is a good bet that our microbiome plays a role in their appetite-suppressing properties. These blockbuster drugs resemble a chemical called GLP-1, whose release from the gut wall is stimulated by our gut bacteria when they break down food. So put simply, GLP-1 is nature's version of Wegovy and Saxenda, and it is the metabolites of gut bacteria that make this compound available to the body. Incidentally, new observational studies suggest that GLP-1 not only reduces how rewarding the brain finds food, but also alcohol, opioids. I'm being told that they're running out of time. Okay. Alcohol, opioids, and possibly even gambling. So, 1 minute maybe? Okay, I'm just going to jump ahead then to what I consider one of the more promising applications of microbiome research in medicine, and that is in the treatment of neurodegenerative diseases. Microbiome science is helping us to unravel the root causes of Parkinson's disease, the second most common form of neurodegeneration. And if you have seen a recent video of Michael J. Fox, the actor, you'll know its symptoms include shaking, hunched over posture, shuffling gait. It's long been known that the disease involves a misfolding of a protein called alpha-synuclein in this part of the brain, and as the misfolding spreads, more brain cells die, symptoms worsen. The big mystery is what causes alpha-synuclein, the Parkinson's protein, to misfold in the first place. So a group of scientists have all converged on one likely culprit, and that is some people harbor a strain of E. coli that actually produces a misfolded protein very similar to the Parkinson's protein. And when that's injected in rodents, you actually, about 2 months later, will see that that normal proteins in the gut lining start to misfold, and you get this sort of domino-like effect where the misfolding spreads up the vagus nerve to the brain, and then you start to see clumping up of the protein in the very part of the brain that degenerates in Parkinson's disease. So needless to say, this has many scientists very excited about the possibility of stopping the progression of Parkinson's disease, at least some cases that we now think may have an origin in the gut. I am being told I must stop. So thank you very much.